Today I'm going to talk to you a little bit about how we use next-generation sequencing to diagnose patients and give them a genetic diagnosis for their bleeding disorders. So I don't have anything to disclose. So a lot of this conference we've been talking about hemophilia, A and B, and von Willebrand disease. And these are the names of the genes that we find variants that cause these disorders. But there are lots of other genes that contain variants that can cause people to have bleeding symptoms. And so today I'm going to focus a little bit away from von Willebrand disease and the hemophilias and talk a little bit more about these. One of the reasons why using next-generation sequencing technologies to diagnose patients with these other disorders is because it's really sometimes not clear what gene is responsible. So we all know that if you've got a low factor VIII level, then it's highly likely we could find a variant in the factor VIII gene. But of course, with some of these other genes that cause bleeding symptoms, we have quite generic bleeding symptoms, and it's not really clear how we can pinpoint the gene that's responsible. So I just want to put the next generation sequence panel testing into context. So to your left, this is what we used to do, and this is what lots of labs are still doing for testing across the world, is that we can look at one gene by PCR and Sanger sequencing to try and identify what variant is associated with a disease in a patient. And of course, if you've got one of these disorders where multiple genes could be responsible, then testing using PCR and Sanger sequencing is often a stepwise process where you're bleeding a patient perhaps multiple times, and often these tests are really not available. And so we've tried to move away from this for some of these disorders where we have more than one gene. In the future, we're going to be whole genome sequencing people in the NHS. We're going to start this very soon, but whole genome sequencing is still a little bit expensive, and it's perhaps not ideal for running for diagnosing patients with disorders where we really have quite a good idea of what we're looking for. We probably expect a variant in 1 or 2 or 3 or 4 genes. So at the moment, and for the last few years, we've been using targeted gene panel sequencing. And the advantage of this is that we can take one blood sample and we can sequence lots of genes at the same time. And this has allowed us to diagnose patients with these disorders. So we've also been able to use this opportunity to develop strategies and pipelines and expertise, and these that we can directly transfer to when we're doing whole genome sequencing routinely in patients in the future. So, as Keith said, I run ThromboGenomics, which is one of several panel tests across Europe and across the world where we're using these next generation sequencing technologies to look at multiple genes at the same time. I'm just going to tell you a little bit about this because I'm going to use some data to illustrate one of my points later on. So what we do is we test about 95 genes in this panel, and we sequence everybody's DNA for all these genes. So we have some coagulation genes, some platelet genes, and some thrombotic genes as well in this panel. So some genes that are responsible for people who have clots, so DVTs and strokes at a young age. So we've received about 2,000 cases from the UK and across the world, and in the cases that we have reported back on, we've found a genetic diagnosis in over half of these patients. So I'm just going to take you through the process that we and others use to try and identify the reason why people have a genetic the genetic reason why somebody has a disorder. So I'm not going to take you through the biology or the statistical steps. I'm just going to try and give you a quick overview. But what we do is we take your DNA sample and we perform sequencing of these certain genes that we're interested in. And I asked the bioinformaticians for a number, and they said that in average, in a Northern European individual, for the ThromboGenomics platform, we see 150,000 variants. So of course, a lot of these variants are common. These are not going to be disease-causing. A lot of them are just differences between you and me. Some of them might cause really slight differences in phenotype that might mean that you've got blue eyes and I've got brown eyes. And we know that for these disorders that we're really just looking for one variant or maybe two variants that we think cause the disease. So how do we get from 150,000 variants down to finding that one or two variants that we think are causing that disease? So the first thing that we need to do is select the gene transcripts. So I've shown you that we test about 95 genes, but genes have different versions as well. And so we have to look in the databases and in the literature and try and pinpoint which gene version that we're really interested in. And once we've decided, we talk about this during the Scientific and Standardization Committee that I'm a part of with the International Society for Thrombosis and Hemostasis. And we use that as a forum for the community to agree on these transcripts. Then we use something called the Locus Reference Genome Database to fix these transcripts. So if in 10 years' time somebody wants to look and figure out what variant they have, they can go to the LRG database and that they can work out which transcript was used to call that variant. So that's really important, trying to fix in this ever-changing genetic landscape. So the next thing we do to get from these 150,000 variants is we need to prioritize them. And we use guidelines that have come from the American College, and there's similar guidelines that are based on these from the UK. So the first thing that we do is we ask, does this variant have an effect? So a large number of those 150,000 variants are going to be sitting between genes. They're going to be sitting in introns of genes, and we really expect these things to be doing, these variants to not be doing a huge amount. So what we're after is missense variants, so variants that change an amino acid, or stop variants, or deletions, or variants that might alter splicing of a gene. They're the ones that we want to find. So we call all of the effects of these variants, and then we have a look to see how rare they are. So of course, if we're looking at a disorder with a frequency of 1 in 10,000 in the population, if one of these variants is seen more than 1 in 10,000 times, then that can't be the reason that that person has got the disorder. So we have these massive databases of control individuals. Some of them have diseases, but we can use the frequencies of these variants in these populations to try and distinguish rare variants from more common variants. And finally, what we ask is, has this variant been previously seen? So of course, the literature and the ClinVar databases, and for us specifically for the bleeding disorders, the EHAD databases are really useful in seeing whether or not somebody else has already seen whether or not that variant is associated with disease. So from 150,000 variants, we get down to 4.6 potentially disease-causing variants. So that's all with the computers, with the pipelines, with the bioinformatics skills and people that we have. But then we need some input to try and find which one of those 4.6 variants are causing the disease. So here we've really focused on for ThromboGenomics, and I think this is going to be adapted for lots of projects elsewhere, is that this MDT is really critical. So I've just got a slide of the whole process, but I want to focus on this multidisciplinary team meeting where we bring the clinical information and the pedigrees, as we've been talking about, of the patient, along with the family history and the genetic data, and we bring these together with statisticians and clinical scientists and hematologists to provide a genetic diagnosis to the patient. So that's our process, and what I wanted to do now was to try and show you the genes that are associated with bleeding disorders that aren't the von Willebrand and CD9 and CD8, and factor 8 and factor 9. So I had a look in the literature. I tried to find some good estimates of how many patients had symptoms that were associated with some of these other genes, and that's really difficult. There's little known about some of these disorders. Sometimes only a handful of people have got some of them. So I used the data that we've been generating as part of the ThromboGenomics cohort and also as part of one of our research programs to try and pull together all of those cases together and give you a good idea of the types of genes that we're seeing and also the numbers of people who are affected by those disorders. So to do that in our database was relatively simple. I wanted to find people with bleeding disorders. And luckily, we code people in a very similar way to coding the pedigree. We code patient symptoms when we're performing our MDTs. So we use this thing called human phenotype ontology, which has a code per term. And so to capture the clinical and lab data. So for clinical data, for example, we have epistasis has a term, menorrhagia, and prolonged bleeding after dental extraction. So not only is this useful, at the time it allows us to turn a text term into a computer term, but it also allows me to go back into the database and pull these people out very easily. We also have lab data terms, so reduced factor 8, thrombocytopenia, for example. So I went into our database and I pulled out 184 patients that had bleeding symptoms. They had a genetic diagnosis that we'd identified. And I put aside the factor 8s, factor 9s, and the von Willebrands. And you can see the distribution of the HPO codes that we have. So the vast majority of people only had 1 or 2 bleeding symptoms or lab phenotypes, but some people had up to 6 of these bleeding symptoms. So I took those top proportion, and what we do is we find 21 genes in this set of people that have 4 or more HPO bleeding terms. And so this was my effort to try and show you guys how many genes are associated with these bleeding disorders and the frequency of some of these genes. So we have actinin 1 and MyH9 with more people than perhaps some of the rarer Hermansky-Putnak disorders. So I've split these genes into different areas. On the left-hand side, we have the factors. So we have the factor 11, factor 7s, and the fibrinogen genes. And the yellow genes are, these are involved in coagulation. The yellow genes are genes that are involved in thrombocytopenia. So the majority of patients will have had bleeding symptoms because they wouldn't have had many platelets. So of course, platelets are the cells which stop you bleeding if you have a cut. They detect activation signals, and they start the cascade of events to form that clot and to plug that hole in the blood vessel. The next set of genes are the genes associated with the proteins that are inside the cell that when the platelets become activated, they're released, and they also cause platelets to start talking to each other and start to become activated. And the final set of genes are the genes that are that encode for the surface receptors on the surface of the platelet. So if these genes don't work properly, if these proteins don't work properly, then the platelet doesn't know that it's supposed to be activated. And so in the same way, a clot isn't formed. So I just hope I've given you a bit of a flavour of the different bleeding disorders. And I just wanted to very quickly talk to you about incidental findings. So, of course, if you're running a panel test where every single person who comes, who's referred to me is sequenced for a lot of different genes, then we might find things that perhaps we weren't expecting. This is a hot topic. This is something that the whole community is talking about. And I think we are getting to grips with how we deal with this. With the 100,000 Genomes Project, there's very clear guidelines of what incidental findings should be fed back to the patient. I think as whole genome sequencing becomes more available to patients, then we're going to have to continue having these discussions. So for the ThromboGenomics panel, some of the genes that we have to think about are the transcription factor genes. So not only do these genes sometimes have variants that cause thrombocytopenia and therefore bleeding, sometimes these variants can increase the risk of cancer. And so we have to think about when we test patients for this, whether or not we are counselling them properly, and whether or not it's appropriate to test children perhaps for some of these. So these are just some of the discussions that are happening, and I don't have any answers. We can also detect aneuploidy with our panels. So of course, we're sequencing with a next generation sequencing. We can look at the read depth that we get across a certain region. So we can detect small deletions and small duplications, which is nice because these can cause our disorders, but we can also detect massive deletions and massive duplications. And we have to think very carefully, and this is why the multidisciplinary meetings are so important, is we have to think very carefully of how we feed those back. And in the same vein, we can also detect, and very specifically for this audience, we can also detect X-linked diseases. So we might have a female that comes to us and she's being, the clinician's asking if we can have a look at her protein S gene because she's got a low protein S and she's had a stroke at an early age. And we might find a pathogenic variant, a variant that causes hemophilia in males. And so we need to think very carefully about how we feed that information back to the patient, if we feed that information back to the patient. There's very clear guidelines out there, and we use the multidisciplinary teams to be able to provide guidance to the clinicians. I just wanted to give you a final example of where an incidental finding was reported back to a patient, and it did matter to that person. So it was a mum, and her son had hemophilia A, and we identified the F8 variant in her son. And we also identified the mum as a carrier. So our normal report would say that this lady had a 50% chance of passing on that variant to her future children, and the children that she already had. We also identified that she had 3 X chromosomes. So this is relatively common in the population. About 1 in 1,000 women have 3 X chromosomes. Often women don't know, and it can cause some developmental problems, but often, yes, like I say, often people don't know. And in this mum, we had 1 copy of the normal variant and 2 copies of the disease-causing variant. So by understanding this, we were able to feed back that she didn't have a 50% chance, a 1 in 2% chance of passing the disease variant onto her other children. She had a 2 in 3 chance. So this is where one of these incidental findings really matters. And this wouldn't have been picked up by old traditional PCR and Sanger sequencing techniques. So I think that's an advantage for whole genome sequencing. But we have to think very carefully of how we deal with these. So I think I've given you a bit of an overview of the gene panel testing, how we prioritize the variants and get down from those 150,000 to that variant that we think is important in that patient. I've talked to you about the multiple other genes that are associated with these bleeding disorders. And I've talked a little bit about the incidental findings. And I think it's really key to think about how we consent patients and how we feed back patients when we find some of these incidental findings. So I've showed you some data today that's from a huge team of people. So thank you very much.